Lunch Session, Session 8. Now, I like to request Dr. P. Saravanan sir to give introduction of the speaker of Session 8, Professor Rita.
Good afternoon, everyone. It is my privilege to introduce our chief speaker, Professor Rita. Singh. He is currently lecturer, Division of Molecular Endocrinology and Reproduction, Department of Zoology, University of Delhi. She was a Ph.D. degree in the year of 1990, University of Delhi. During this postdoctoral research, she served as a member scientist 19th Indian Antarctic expedition during the year of 1989 to 1990, funded by the Department of Ocean Development in Delhi. She has served as a CSIR Research Associate at the Center for Cellular and Molecular Biology at India and scientist in various Ph.D., SSASC, and the Indian Institute of Science, Bangalore. She served as a lecturer in Delhi University and All India Institute of Medical Sciences. The research area includes studies molecular development in polycystic ovary syndrome, crosstalk between signaling pathway in IGF-1 and FSH in granulomas proteomics. Here, she worked in 17 research projects funded by DBT, DST, Indo-US, CSIR, UGC, R&D, DU, UGC, and DST, DU Plus. She has received many awards. A few of them include Science Merits, Scientific Science Merit Award, UGC Grant Commission Fellowship Award, DST Young Scientist Award. And he also many academic bodies like American Association of Cancer Research, USA, American Society for Biochemistry and Molecular Biology, USA, Federation of American Society for Experimental Biology, USA, Indian Society for the Study of Reproduction, the Fertility Society for Cell Biology, India, Society for Comparative Endocrinology. Also, she delivered an expert service as a mentor, moderator, reviewer, nodal officer, represent in many prestigious institutions. On behalf of Sri Shankar Arts and Science College, PG and Research Department of Biotechnology, I welcome you.
And now for the last session, I would like to request Professor Rita to present the 8th session on Recombinant DNA technology, therapeutic proteins.
Ma'am, thank you very much for such a kind introduction, and I really feel thankful to Professor Murtidar and all the organizing team for organizing the workshop where it's a very different kind of workshop. That in different people with a different kind of presentations. Giving my gratitude to all of you, I start with my presentation. Is my presentation clear to all of you? Yes, ma'am. Okay. So today I'll be talking about, as you all know, on the therapeutic protein, and I have particular slides where I'll be talking about the proteins and the kind of technologies which are used for producing these proteins and the applications which are demanding these kind of protein drugs. So, to and these protein drugs are, we must understand that these protein-based therapeutics have been highly successful in clinic as well as in the scientific world, where they have really helped scientists to discover more and more in the field of biomedical sciences, as well as helping the clinical people to use that. And slowly we will unravel all those things. So if you look at that, almost 120 genuine and similar kind of other therapeutic proteins have been approved by Europe and USA. And if you see the latest difference between 2018 sales of these therapeutic proteins, which costed around US dollars 93.14 billion in 2018, to the current sale, which is 112.17 billion, it's almost 20.43 jump. And this is happening every year. There is more and more, sale of such compounds. And we all know during COVID, so many different kind of antibodies which were being pushed into the market, though maybe without the trials. But there are several other, monoclonal antibodies, which they account for almost the sales. We know these antibodies are used for several different diseases. So if you look at various different kind of therapeutic in their market in different parts of the world, you see USA is the biggest producer of hormones in like insulin, fusion proteins, recombinant vaccines, and monoclonal antibodies. And Europe is the next major producer of these proteins. Here, if you notice, colors in this pie chart, they define the kind of expression systems they are using. USA has more, money spending capabilities, so they are using many mammalian cells based systems, whereas in Europe, you see they are using more the expression system. All these things I will explain in a little while. But then there are, other countries which use E. coli or the bacteria-based expression systems, and they have almost 28% of. And these may be hormones, fusion proteins, vaccines, antibodies, and growth factors, which human consumption as well as for research. Research is a very big market, like the kind of research I'm doing. And the stem cell research, you have seen several lectures on that. So in China, if you see, they have certain other expression systems. So what are these therapeutic proteins based on the molecular types? You can see that there are antibody-based drugs like Fc fusion proteins. Now these are also, being made in a, size smaller than big proteins. So these are also called nanobodies because of their use in the therapeutic as a therapeutics, but otherwise they are peptides which are, being discovered and being used because peptides will not, initiate an immune response in our body. So the peptide therapeutic drugs are being put so that immune body into the response. Then you have, another very important category of proteins which have helped several because, there are many diseases and genetic diseases where one needs anticoagulants and blood factors. Then you have various, proteins like bone morphogenetic proteins, and then engineered protein scaffolds, enzymes, You have heard the lectures on enzymes, how important they are for the people who have bad digestion or they have genetic issue in enzymes from pancreas or from the digestive system. Then you have growth factors, hormones, interferons, interleukins, and thrombolytics. And you know, in the patients with strokes and other things, how important it is to have some of these. And so let us see some of these proteins, which were, the initial phase of production and patients. Before 80s, when we did not have good supply of insulin in India, several diabetic patients, patients who developed type 1 diabetes later in life, their insulin production just stopped in the old age. So they died in and the supply of insulin was not that good. But then once the recombinant insulin was produced, there were several who could survive. There were several old people who could live longer. And this really helped in diabetes mellitus. Then interferon alpha, this is very useful for chronic hepatitis C. Interferon beta, which is used for herpes and viral enteritis. Coagulation factors 7 and 9, they are used for various hemophilia conditions A and B. DNA 1 used for the treatment of cystic fibrosis. Antithrombin 3, it prevents the blood clots, which is very important in old age as well. Interferon B for treatment of multiple sclerosis. And then there are several individuals which have a problem with their growth hormone, sometimes genetic. So these hormone, recombinant growth hormone, can help them survive. And then you have plasmin activator, which is very important for the treatment of acute myocardial infarction. So now, proteins, if you see, when I tell my MSc students, I tell them that protein, I ask a question, what makes us living? DNA can be found in a dead sample. So what living is the protein? So for the first time when they hear this, they feel very surprised. Protein is a protein. How does that? When they realize that it is the protein which senses, it is the protein that acts, it is the protein which has the capability of activity. It means it's alive. It is doing the functions of everything which is coded in the DNA. So that is why it has become very important and imperative that we understand proteins and then go about treating the diseases. Because it's not case that the expression of the protein is there or not there. It means that if gene is different, is not the only reason for disease. It may be that the gene is expressing a protein which is not processed in the body properly. So what in most cases the disease most. But the most important challenge now in the current era is that to understand functionalities, the protein, how it functions by even a small modification to this protein can cause enormous amount of difference in activity. So that is why the science of protein expression systems developed. And then this slowly we will see how it grew, but the first protein was expressed in 1976 76 in bacteria, the Escherichia coli, we call it E. coli. So Genentech expressed hormone protein, which is somatostatin, which is a very important protein. And this was expressed in, and later on, the insulin was expressed in 1980, 80s, and then it was in 82 that it was finally put up to the world. So when we look at these recombinant therapeutic proteins, why we would do recombinant? Why we could just isolate from the tissue sources, like classically we were studying and doing during our PhD, did that purifying hormones from pituitary of animals, or we can do it. But then problem is that we want something for the consumption of human. So we need human proteins and get human tissues to isolate the proteins. Even if you get some tissues, but then that much will not be enough for the consumption of the kind of population one has to deal with these particular defects. So that's why the recombinant technology became very. But most people and the curriculums are also still deficient in this coursework. This is not a part of anywhere I see is never taught in any of the master or bachelor classes. So that is why it is very important that we understand what we are doing. But it's a very simple science where all these things were developed, like these proteins which were as you know, enzymes and other regulator proteins, or these can be targeting drugs, vaccines, and diagnostics. So then, how do we understand how these, proteins we to understand their chemical characteristics, because we want them to be functional for the purpose we are, expressing them. So if you see based on their molecular mechanism of activity, you see monoclonal antibodies, they have the property of binding to a target, that's an antigen. And then you have enzymes which may bind through the covalent bonding, or there are other proteins like serum albumin and several other which may act differently without any specific interaction. So now these, if you see, these proteins can be used to substitute a deficiency, or a person, the protein which is made abnormal, so it's not acting. So if you substitute this particular protein, then the person can survive. The most important examples are very well known to all of you, like insulin, albumin, and other coagulation factors, and then they can augment an existing pathway. Say a particular pathway in the body is not working properly. In case of infertility patients, sometimes this cannot be called infertility, it is called subfertility. It means that the person has the need to be fertile. Say, for the example, hormone is not functioning properly, maybe it has developed some problem. So people go F, and then they take treatment with FSH and LH, and then they can have their conception properly. So similarly, in case of certain immune diseases and some certain other pathways, what happens is that you want to, and then the person can live a very healthy life. And then you have certain other novel functions which can be there, which are new discoveries in the over the period of time, where people maneuver things in such a way that the person can, in a particular situation, they can have things like, if you see in case of cancer and various it's like the organ transplantation, or in certain diseases like thrombosis and all, you need immediate, can't have you do injection and wait for the reaction. You need immediate interference, like antagonist kind of. So these proteins which are made, they are made with an idea that they can be very efficient antagonists to a particular kind of signaling going on in the body or interactions going on in the body of a diseased person, like pulmonary disorders, need immediate action. So those kind of protein or peptide antagonists have been made, and the antibodies. Now what we are doing is they are making very small, as I told you, nanobodies. So these are very small peptides.
It's 11 o'clock.
These, what they do is that they are tagged with certain other drug and they are used as the delivery agents. So the big portion of the antibody which binds to a particular tissue or an antigen in a particular tissue, that they are used for targeting. And that is how cancer as well. So they are tagged with a particular drug which will go and only attack the tumor. So now there are other therapeutic vaccines which are based on the recombinant technology, and they can be towards any of the deleterious infectious agents. And then obviously, autoimmune are on the rise in all the countries, and you see there are patients suffering with arthritis and many other things. But now several kind of drugs which are on trials, and specifically for cancer, where through specific targeting as well as the efficient antagonism of the signaling pathways. Because why we are needing these protein based for all these functions is because these diseases are being caused by the proteins. These are being caused by malfunction in the protein infections inside the cells, whether it is a tumor, whether it is a bone joint infectious situation, which is difficult to handle. So in such situation, have a particular protein-based therapeutics. Because many of the organic compounds have been used for the kind of therapeutics, and all are familiar. These diseases could not be tackled with them. But when people use specific, portions of immunoglobulins or a specific portion of which is a small peptide-like structure. So now what is it doing is it is typically for the human, amount of human protein itself. But now this will go and bind to overexpressed protein inside body and will not let it act in the body to generate more immune response. So these are like something which we are idiotypic antibodies. So instead of a complete antibody, what we do is we just make a small protein in a recombinant manner, then inject it as a drug, which will go and stop that particular over expressing protein in the natural environment, which is causing the autoimmune disorder, or an antibody which is causing an disorder. And obviously, there are so many diagnostic kits which are made nowadays, and you can't afford to do it with the isolated hormone. You need a lot of manufacturing and other things, and it is very easy to make various kinds diagnostic kits. You need the antibody, you need the protein. So these proteins can be in some express big protein, as well as the protein drug, which can be smaller in size, which have better penetratable properties, which can easily be targeted, and which can have a cell entering something like which can enter the cell. The cell membrane is very, very impermeable to proteins. So how will you make a drug to enter a cell? So that is still a challenge. That's why a lot of response for the tuberculosis, where the infection is inside the cells, and one has to find ways to efficiently handle that without being, having so many side effects are there, the drugs which are being used at the moment. This is again the list summary that various proteins, how they are being used. I have already explained this. And these various manufacturer companies which, made these proteins and they are selling these proteins. So this market has really pharma companies earn about discovering this kind of or making this kind of recombinant proteins. So why express or P5 means? Because we, uh, uh, when you don't have a source that you just isolate a gene. You can you have to purify protein, and then only you'll be able to understand what this protein is doing, what its sequence is, whether make antibodies. Even if you see the antibody market, or even for research, when I purchase antibodies, they're so expensive, and people make proteins. It is so easy to have a purified protein to make specific antibodies. So I will explain to you that purification is also a very different term when you are making a recombinant protein. You don't have to go through an elaborate process of. But when you are doing research, you want that your protein, which is a perfect sequence, so that can easily be identified by mass spectroscopy. If you have then coats the protein, you may want to express and purify the protein for other reasons that we have already explained. These are various kind of applications for research and education. And the last which I've explained so far, but others can be for various kind of understanding of the proteins, because that is also part of developing the tests when you want to do analysis, enzyme activity, interaction partners, its biochemistry and functional studies, so that you can conclude that protein you are expressing is perfect for what it is known, or you can discover new protein and you can characterize by all these technologies easily available, and you can understand that. So just for the students, I had put this simple slide where that the RNA is synthesized inside the nucleus, but then it comes out to the cytoplasm, and then it makes proteins. But then you see, when within the cells, there are several major control points for expression of protein. Why we have this whole cassette of translation in the cells? It's not that protein is expressed and it stays in the cell. The protein immediately gets degraded after performing its function. Other than even the structural proteins, the skeletal proteins, everything is recycled or replaced by the new synthesis. So this cycle as protein synthesis within the cell goes through from mRNA getting to the cytosol. Now, once RNA is there, the coding portion will be the messenger RNA. Now, this messenger RNA for a particular protein, when it is expressed, it will not be cytosol. It will get degraded soon after it has gone through a translation process. So there must be a cell where a particular level of protein is produced, and then that gives a signal that now mRNA is no more needed. So they get degraded. So there are controls there, and then there are translation controls. It's not that if there is unlimited translation of cells in the eukaryotic cells, then it will just cause again the problem. So therefore, in a normal cell, the protein is just made for this, and then the rest of it is translation is stopped. But now, if this protein is increased in its in cancer, then you can know that overexpression can cause, like Wnt5 overexpression causes, one overexpression causes cancer. So various proteins, cancer is a very easy example for showing the so when we want to express the protein in the laboratory on the bench, then there are several different things which you will have to understand and design. So now the first point, engineering of the protein construct. So you have to decide whether it is the entire protein you want, or you want just a particular domain, or you want to make a rather a mosaic protein where you want to connect two different proteins and then want to make a construct. Many people have done that, where there are alpha and beta subunits like for gonadotropins, or there are receptors where there are like insulin receptor or any other, where there are two subunits people have combined and tried to construct a single protein or understand their functions. Then the next question will come, which organism to express the protein? Now, if protein is of bacterial origin, you can express it in bacteria. But if protein is of the eukaryotic origin or human origin, you can still express it in bacteria. But then the bacterial origin proteins or the translation does not get completed in bacteria, because we have several post-translocations in eukaryotic system. So you have to then go to yeast or insects or to mammalian cell lines. So this particular I have put in a sequence where people follow that because of the cost involved in the getting the proteins. So bacteria is the least expensive and time consuming, and mammalian cells are most expensive, need more time. So that is why the sequence is from bacteria, yeast, insect, and then mammalian cell line. So now if you need to express a fusion protein or require codon optimization, so these things I will explain to you certain basics. But then for that, let us first understand the various kind of expression systems which are used to express the proteins. As said, first and the foremost is bacterial expression system, most easier to do as well. But the thing is that there you can make eukaryotic proteins without any post-translation modification, but some proteins like insulin were easily made and functional, so it was very easy. So you have in this plasmids and phages. This I will explain to you. Then you have yeast system. There again, in yeast you can have plasmids or you can have yeast artificial chromosome. So these are talking about each expression system and the kind of vector, the expression module, the machine for expressing the protein. So these are different cell lines or cell types. So in insect cells, you have the vectors which are called bacula plasmids. Then in mammalian system, when you want to express the proteins, you have various different kind of viral vectors. And these viral vectors are also used for gene therapy, but then these are used for research as well as for transfection of a particular gene into a mammalian cell and its overexpression. So various different viruses which are used are F40, vaccinia, adenovirus, retrovirus, and different cell lines which are used are CO, which is the Chinese hamster ovary cells, and hex cells. These are two popular cells which are the expression of these proteins. So now these, most of the time, the CA cells view much better expression, higher expression, though both are very, very And these kind of cell lines are used in the research I'm doing. Maybe in the end, if I'll have time, I will tell you how we are using this whole technology for the finding of gonadotropin signaling and to discover or to plan a therapeutic protein, the kind of hormonal imbalance which is there in PCOS cases. So choice of the expression host. So let us the advantages and disadvantages. Now, as we have already discussed that in bacterial system, it's all good, easy for a person or technician to handle it. But sometimes there is problem of protein solubility. The expressed protein goes into bodies. Now, human bodies are kind of structures which are very difficult to dissolve in normal solid use for protein solubility. So one has to use harsher chemicals like urea and other things to solubilize, and that may not be good for the therapeutic proteins. So one has to go to the next level of, and then of course the modification happen here. So therefore, in such situations, one has to go to eukaryotic expression system, which I have shown you various different kinds. So you will have all the advantages of protein solubility, post-translational modification, but then there will be problems like low yield, and it may be time-consuming and expensive, of course. Then you have the isolation of protein from natives, which I discussed at the very first point. In that case, you will have the problems like you have to go to the slaughterhouse, you have to use varying blenders, and then you have to see that in the end your protein is pure. That is another important thing. May not be very expensive, but then it is labor intensive and it does not assure you white protein. Absolutely no other contamination, because sometimes there may be some protein-protein interactions, and there are some proteins strongly bound to the protein which you are editing. So one has to, one has several things when you want to use the protein for antibodies. So when you sell the antibody, it may have another tagging antibody which may have more side effects. So like that, there is some, so it may be okay for various kind of research purposes to confirm the protein structure. But mostly people went into the eukaryotic expression systems, and based on the quality and the quantity of protein required and expenses which you can generate, bacteria, yeast, SF9, insect, HeLa, and tissue. This is the kind of hierarchy which is used. So when it comes to the production of recombinant therapeutics, companies use a simple model. They generate a gene which can be generated with a PCR, and then you have the full length DNA, means it is the generated from mRNA. That means a coding portion of the final mRNA, processed mRNA, that this cDNA is. So when you insert this DNA into expression vector, I will explain the expression vectors because that is something very important. You just, it's like in nothing very technical, just one has to cut paste kind of a thing, and sometimes you need glue, that is an enzyme. And then once you have this whole wire ready, kind of a wire ring of things, then you just put it into whichever cells you have. But the most important thing is clones. As you all know, these vectors or plasmids, they have various kind of markers for these things. So I will explain to you. So once you have the vector which can express So you have to decide what kind of construct you want, a bacterial construct or a kind of mammalian construct. So there are slight technical differences in these because of the expression of the protein requires additional components in the plasmid. This I will do later. I want to show you this first, the vector. So now when you want to express a protein and when you just wanted a vector with the DNA for sequencing, these are two different plasmids. Like PUC is used for cloning of a gene and sequencing and other things. But when you want to express a protein, this vector, which is a circular structure, it has certain components and there is nothing very complicated in this. What your sequencing had was multiple cloning site, and this particular vector where you want to express the protein, this will have certain other components. Now, number one, you have a promoter. So even in cells, when a protein is expressed, there is a transcription factor which binds to the promoter, and then this promoter activates the transcription and further goes the translation. So here again, you need to have a promoter which will activate the transcription of the gene you have put in, and then that mRNA will be then expressed into a protein. But the most important, how this protein will be isolated. So for that, what is done is in this construct, there is another protein which is put into, which is glutathione as transferase. For example, this is GST. Now, next to the promoter is GST, and then is the multiple cloning site. Now, here you will put your gene. Now you will have certain, I will show you in the next slides, you will have certain cutting points here with the enzymes, which are called endonucleases. So now what will be done is that you see, you have this marker protein, which will make a fusion protein with your protein. Say, you are expressing hormone FSH. Now you have put the gene for FSH here and GST is here. Now you can isolate FSH and glutathione as transferase very easily by affinity chromatography as a pure protein. Now the fundamentals here are that how do you check that your this thing is protein is cloned and the gene has been properly cut pasted. So here you have this marker, which what will do is that you have in the other vectors also like selection markers. So they happen is as soon as the gene is going into here, it is going to disturb that enzyme. Like you have like Z marker. So that expresses an enzyme. Now this enzyme, what it does is when you put a substrate into this culture, enzyme will produce a blue color with a substrate called XGAL. But when your gene goes into it, this is interrupted. So all white colonies will be the ones which will have your cloned construct. So now this, because the beta glycosidase will, cut the this XGAL, which is an artificial substrate for it. Otherwise, beta galactosidase does cleaves the lactose into the. Lactose is a disaccharide, cuts into galactose and glucose. But here we put an artificial. It is also called a blue enzyme or blue indicator enzyme. So when you put a gene here, this enzyme sequence is interfered. So it is not active in your cloned sequences. And when the protein is expressed in the presence of xgal, you put IPTG. So now you need IPTG to suppress. Now there is a lack repressor here in the normal this thing. If you want to express your protein, you will have to put a chemical called IPTG, or it can be certain antibiotics also for which suppress repressor. So in normal condition, this repressor will be expressed and it will not let the beta galactosidase get expressed. But when IPTG, now this is repressed and this whole cassette will work. And you will have the selection of the colonies, bacterial colonies, where only white colonies means that your protein of interest is being expressed. So then you take that white colony and then you streak it on the agarose plate, and then you have the this thing cloned. So we go back and see why plasmids are cloning vectors. Now these are very small size, just few kb, 3 kb, 4 kb, like that, and they are circular. Very easily you can see the structure of. So what is the structure? It is nothing but a DNA. DNA with coding sequences, like you have to express each one of these components. So these vectors are easily available in the market. What you need is a gene of interest. You have to understand how expression happens. So same knowledge, what is happening in the cell is used here. You have a promoter and then certain markers. So then you also have ampicillin marker or any other antibiotic marker. So what is happening there is that you put the antibiotic, only those bacteria which have the insert will survive, rest all will die. So that is another way of selection. This antibiotic selection is also done for the sequencing vectors, where you just want to have more of DNA. You want to amplify your gene of interest, and then you want to do various studies like epigenetic studies, methylation, and other things. So you need more gene also through these systems, but there you will not have the expression cassette like the specific promoter for your gene and other selection markers. So, and then the one of the important thing which while selecting the plasmids, you need to give a thought. There are certain plasmids which have very high copy number. Means if you transect bacterial cells with your one type plasmid, it takes just 10 copies inside the cell. But there may be other plasmid types, so which will make much less copies. So you want more to be isolated from 100 bacterial cells. So then you need a plasmid which makes more copies. So like Puck and all very popular this thing vectors, or this PGX also is very good vector. So you can use these bacterial plasmids are very easy to maneuver in the laboratory. It is just a mixing of chemicals and the but when it comes to expression vectors, the plasmid is frequently engineered to 3 sequences, as you said, that act as enhancer and promoter regions and lead to efficient transcription of carried on the expression vector. Now these expression vectors must have expression signals such as a strong promoter. Promoter is also different for bacterial and for the mammalian system. I will show you. And then and that will also depend on your gene. But then the common things which you all know, a strong termination codon must be there. Adjustment of the distance between promoter and cloned gene has to be optimized, and the insertion of a transcription termination sequence is very important. But one of the important things is a portable translation initiation sequence. Now this is a sequence where ribosomes bind. So this is something specific for eukaryotic or bacterial system. So you have to see that the translation initiation signal has to be appropriately put in the vector. But then nowadays everything is available on the shelf. That means in the market you just ask them for a search on Google, need this vector with these things, and there will be a name. You go and just buy. But initially, when scientists were working on it, they had to see to it that all these things are there. So these expression systems are also used. Very easily you can by PCR you can introduce some modification in the gene sequence, and then you can study if I have this particular polymorphism in a disease condition, and if I put in this gene, will it function properly? Will it have its enzyme activity? Will it changes in its protein interactions or in its signaling? So these plasmids, they are very, very easy to work with, and they are not like working with RNA, which is just gets degraded by if it gets even your smell or touch or anything, because everywhere there is RNAs. So these kind of expression systems are very subtle and very stable, so you don't have to worry for anything. So when you see, this is a list of various kind of vectors which are used for and their various kind of copy number is given here. If you see, Bach vectors have the maximum copy number, and then you have the blue script vectors. These are also used for the expression, and then PGEM vectors, they are also having high copy number, but PTZ vectors have very high copy number, that is more than 1,000. So most commonly used are PBR322 and derivatives, but many times these do not have very high copy number. They may give better expression sometimes. Then you have several other kinds, but they have very low copy number. Then there are another kind of plasmids which are called cosmids, but difference is just that they can take a bigger size of your gene, several kbs, 40 kbs or 30, 20 kbs, like that. So these have normally with the very big gene sequence, these cosmids have not so high copy number. So copy number is that when you transform a bacterial cells with the plasma, how many plasmid will be there in bacterial cell. So that way it can tell you that how with one culture you can get a higher yield of gene than doing a large fermentations, and so incompatibility of plasmids can be there, that not all plasmids are coexist in the same cell. Plasmids which have the same replication control functions are incompatible and are assigned to the same incubable group. So these are the technicalities which you may not bother at this time. But then what it says in a brief, this thing is that when you are using a particular plasmid, you have to see that where and what you are planning to do. But that is also not complicated for the kind of research which we are doing in our biological systems. We can use any one of them. There is and if I tell you, the first three are the more advanced, and only a very special research this thing that people use. PGX vector, which I showed you, is the most preferred vector which is used, because it's very easy to purify the proteins. With the agarose column, which is ready to use, is available in the market. So just put your gene amplify it by this thing. But the most important thing one has to see while cloning the gene for expression purposes is that you have to have a proper reading frame, or that we have 3-letter code. So now when you put the sequence in a particular vector, you have to see 3 reading frames can be there. So one has to understand this reading frame thing when you are cloning a particular gene sequence. So that is where one has to see the codon optimization. That ultimately you get a protein in the right ORF. So then the protein which is generated will be of the sequence which you want. But if your cloning makes the ORF is, then it may read it differently and it will read any 3 codes further down. So that may be an unsuccessful attempt. So that's why in a simplest way you have the clone, which is so major things which you have to take care is that gene dosage, copy number that we have seen, the ones which are used. Blue script is very easily used, or level of expression what you're getting, and the um, compartment to harvest from. So this I will show you another slide, little advanced information further down. Then tags of purification, as I told you, there may be PGX has the GST protein tag, but one can have several other tags, like his tag is there and other. And codon usage is something which I want to explain to you further down with another slide. What does it mean? This is a very important concept. So certain concepts I'll be discussing a little later. So then the purpose of expression, what do you want? Are you a scientist? Are you a person in the industry? Or are you just doing certain analytical level? So all these things will make you make your life very easy if you define yourself, because for scientist you can use some of the very high copy number plasmids, but for some other you need certain other things. So these certain things which we have discussed so far. And then, so now this is codon optimization, which we have to see that if it is a mammalian protein expressed in bacteria, so you have to understand that in bacteria, certain codons are there which are taken as a particular amino acid. So you have to see that in your sequence, you do not have any codon which a bacteria will get confused. So that is why this codon optimization has to be there. I will not have a lot of time to explain these things through the biochemical slides, but this is something which you can easily see. Then the cloning method and all we have discussed so far may require expression as a fusion protein, as I. But one of the important thing is that protein may require co-expression with molecular chaperones. Now let us take an example of this particular thing. In the cell, most of the steroid receptors, like estrogen, progesterone, any of these steroid receptors, they are co-expressed with molecular chaperones, that is shock proteins, and they are always bound to the chaperones if they want to maintain their structure in a proper manner. So, but then there are some other proteins which do not, or they are not able to, that particular portion of the protein which you're expressing, it will not be able to stabilize itself until you co-express another protein. So molecular chaperones are other proteins which are cloned in the same setup. They are expressed and they help in the co-expression of these proteins. Then, of course, you have affinity tags for purification or protease cleavage site, so that you can cleave off the particular tag from the protein and use it as a purified, because you don't want your GST or tag or anything. So you have cleavage sites. And then, of course, the purification and everything is very fast. So you see, when we express protein, another important thing which comes. Now, this important thing is the fact these proteins are as sensitive as we are. So some proteins may get inactivated or may not get, organized or may not express properly in bacterial cells, which are cultured at 37 degrees. So like there is one type of bacterial strain, E. coli strain, which is DH5 alpha, when we transform and then we do the culture, we do it at 37 degree. But there is always a problem when we are doing certain signaling molecules, certain other sensitive molecules, or there are certain proteins which are like disordered proteins. Disordered proteins are the ones which they have no defined protein structure when they fold. So when they are expressed in such situations, they degrade. So different type of proteins in our cells and our body, and those are the proteins which are very critical regulatory proteins. So when you want to study those things and you want those proteins, you need little more expertise in this. So now what happens is we don't, if you are facing a problem that your protein is getting in a bacterial system or it's going into some other inclusion bodies, then you change the bacterial strain. So now this BL21 strain, which is DE3, what happens is this also has almost similar stuff, but what happens is this particular strain is the protease deficient. Okay, so now your protein will not rise, one thing. And second, this bacterial host BL21 can grow at 30 degree temperatures. You don't need 37. If you put the transformed DH5 alpha strain of bacteria at 37 degree, it will grow well. But if you put it at 33, grow well. But then BL21 is preferred for protein expressions because it is one thing is that it is protease deficient, but also it is that can grow, divide, and give you a good cell mass at 33 degree or even 30 degree. Then there is another strain which can be used. So like that, depending on your protein, some of these D3, strain has a leaky expression. So there is another one, D3 underscore P lysinase. So these are further modified to have, sometimes their protein, the expressed protein is toxic. In that case also you need another. What I'm trying to show you here is that there are different strains for expression of proteins of the same bacteria E. coli, and they can be used for your particular purpose, and you use this IPT for inducing the expression of protein from these vectors. So you know that now there are specialized bacterial cell lines for protein expression. You see different companies have taken out various different kind of cell lines, and you can have a look which can make you decide that the ones which are more stable mRNA, and there are codon usage deficiency, it can overcome. Then you have the tetracycline induction, expression of toxic proteins is taken care of, decrease in leaky expression. Like this, now different companies are giving you different kind of but the more important is the origin of replication and other things which are kept constant, and some certain things are so don't use it concern okay, luckily I have the slide. So here different you see different organisms. If the codon usage, these are the codons used for making arginine. This is also used for arginine. This is used for leucine. This is for non-isoleucine and proline. So if you see different organisms, which here you can see this is E. coli and Homo sapien. Like if you just compare these two things, you see this code is used more by Homo sapiens than the bacteria. You see, even this code is used more by the Homo sapiens than the bacteria. And like this, so depending on, so this code is used more by the Homo sapien than bacteria. So this variation may affect the sequence of your protein. So similarly, if you see the other expression systems, so you see there is different kind of codon usage. Only this particular codon is used so highly for this, not this one. But say if this one is not there in your sequence, only this one is there, then what happens to your protein? So that's why one has to see that, like if this for leucine, this is used by this particular expression system, or in Saccharomyces, this is used at 21.3 fold more than the others. So if this code is not there, and if this code is there for arginine, then you see the difference in the protein, what will happen to yours. So one has to, while looking at the sequence, you can put this particular code instead of this code. So wherever you think arginine is coming in your sequence, instead of putting this code, you put this code. So that is what I'm trying to say in this slide, that you have to see which code is important for your protein and which is recognized by the expression cells, which cell line you're using. So this is called codon optimization or codon usage in a very specific manner. So what will happen is that the gene which is expressed into a protein will require little more thought on the expression of your gene sequence to protein, so that you can do it on your computer. So like this, when you know much, so you don't want to put a kind of AGG somewhere if you have in your sequence, and you're expressing it in cynorhabditis, or you're doing it in any human cell line, or doing in bacteria. So each expression type of protein, say if you're doing a mammalian cell culture and you're studying some signaling, there your protein will have more of different sequence than depending on the code. So when you're deciding a gene sequence, you have to see that your codon optimization is done. So this is another vector which is very commonly used for protein expression, which is a PET vector. So now here, if you see all these components I have explained to you, now if you see this multiple cloning site is the one which you are going to use, and it has several sites which are showing you the different enzymes. So if you see in this kind of schematic diagram, you have all these start codon, stop codon, and then all these things. But one of the important things is where your ribosome bind is going to happen. So in bacterial system, these are the sequence which are called Shine-Delgado sequence, which is there for the ribosome binding site. That is the start of mRNA. But when it comes to and when it comes to human expression vectors, now there for this, your ribosomes will bind. You will have a different, sequence, which is called the Kozak sequence. So now these Kozak sequences, they are specific sequence surrounding the start codon. So these are for efficient initiation of translation. So there we had the Shine-Delgado sequences, but wherever, if we are using a mammalian system expression, you will have to have Kozak sequence. So these are the things which are important, which you must understand, that you cannot use a bacterial vector and you say that, oh, I want to use this for the mammalian expression. I will put it in the mammalian cells, and then you say your protein is not expressing. So you can take certain vectors which are common for both mammalian as well as bacterial system. So what people do is they choose vectors where you have both the options, where you can, these are called kind of shuttle vectors. You can make it bacteria, and then you can also put it in mammalian. So that is something which one has to understand. I hope that makes it clear. So these the for the the whole process. So these are certain important components I thought I should. So this is the just explanation of how one does the animal cell culture or the mammalian cell culture. Now here the concept I want to explain is that these, when we do the we put the vector into the bacterial cells, then it is called transformation. But when we put a vector to the mammalian cells, it is called transfection. So if I would have been physically there, I would have asked that question, but now I'm just doing monologue. So now when we do mammalian cell transfections and put the mammalian vector into the mammalian cell, then again there are two things which are there for your selection, whether you want to do a transient transfection or a permanent transfection. So what does this mean? So you have put this, so when you are doing transient transfection just for a study in a week, then what you do is you take a vector, mostly these are viral vectors or vectors which can, or if these are other shuttle vectors or which can be used for bacteria as well, we use the transfection reagent for that. So, or we do electroporation and which gives some vibration, electrical vibration, and it makes holes in the cells. But for bacteria, we just use which makes holes in the bacterial cell, and the vector enters into the bacterial cells from there. It's called transformation. But in case of or the mammalian cells, you use the vector as well as which will be able to make this vector enter the mammalian cells. So now if you see the differences between transfection and transformation. So transformation word we used when we put a vector in the bacteria, and transfection when we put it in the mammalian cells. So transfection means that process of deliberately introducing naked or purified nucleic acids into the eukaryotic cells, introduces foreign DNA into cells, uses co-precipitation, liposomes, electroporation, gene gun technique, and microinjection for gene transfer. In case of bacteria, there is uptake and incorporation of exogenous gene material from the surrounding through the cell membrane, because there are holes which are made by the same calcium phosphate method. Then the foreign DNA is introduced directly into these plant, yeast, or bacterial cells. Use transformation for all three types. And then you can use the similar methods as for human, but these are this is called chemical transformation, electroporation, or particle bombardment. So in case of mammalian cells, what does it mean when we say transient? In their cytosol, had said, yeah, here. You see these plasmids are here. So they can harbor plasmid in cytosol, and this is a natural process for bacteria. So they are self-replicating, and so they can make their own replication and increase in number. But in case of mammals, it is slightly different. So now what happens is when we do transient transfection, the vector has gone inside the cells, and it will use because we have the Kozak sequences, it will use the ribosomes, and it will use the gene, and then it will make the protein. So they don't need any selection procedure like the way we do it in the bacteria. then that you can have like sometimes we use certain antibiotics for even triggering the expression of the protein. So like that may be there. So one has to understand that antibiotic there is not for selection but as a marker for initiation of the expression. So both DNA and RNA can be used for transient transfection. Now, high copy number of the transfected genetic material results in high level of protein expression. So typically, they are harvested within 24 to 96 hours. So that's what I said. Within 1 week, you finish your experiment. Generally, not suitable is using vectors with inducible promoters. So when we do these kind of transient transfection, these are done with the vectors which are expressing the proteins constitutively. You just express, you transfect, and then you in 12 hours, you the expression of the proteins. These are not induced. There is no IPTG, there is no inducible chemical which is used in these situations. We just transfer and then culture the cells and do the treatments, and we expect that the protein is expressed. And once you trigger, say I express FSH receptor and LH receptor or insulin receptor, and then I put any of these hormones and I check the downstream signal molecules like cyclic AMP you can assay, or you can check for PI3 kinase activity. So you don't have anything like IPTG or any inducible chemical. Nothing is used in mammalian system. You just put the vector, it will express the protein, and then you just harvest the cells and isolate, and you can isolate the protein. Now, in case of these mammalian systems, you can, what difference comes when we call it stable transfection? Now, in case of stable transfection, when you do transient transfection, you now go through the generations of these cells and select these cells which still do the genetic material which you have transfected. So over the time, when you say, for example, you have CHO cells and you have transfected them with the particular FSH receptor gene and a vector, what will happen is when you passage these cells, say you again nice cells, again you next passage, there will be several cells which will lose the plasmid. So, but then there will be some cells which will always have the plasmid. They will be expressing the protein which you desire to. So in that case, one need to select by screening of the stable transfectants. That means you do the kind of, you do screening for the monoclonal antibody or for the bacteriophage or any other things. You do a single cell, dilution of the expressing this thing cells, and then you get out a clone, which is a cell clone. So now stable clone means what? It means that single or low copy number of these stably transfected cells, they are expressing the protein, and they are maybe low level, high level, but these are the cells, they have got this gene integrated into their genome, and now they will be, all the cells in after election will always have this overexpression of this particular protein. So this is just the IPTG which I told you, which just inactivates the lac repressor and let it this thing which is suppressing the beta galactosidase, it is presses that, and the expression starts, the promoter. So this is another example which I had given, that sometimes you need a heterologous protein to be expressed along with the regulatory protein, means the one which you are studying. So you will have two proteins simultaneously expressed in a system, like we talked about the chaperone proteins. So protein science is a bit of complex science when it comes to factors and regulatory proteins. So, but then understanding that is the most important thing to handle a disease or to find a therapeutic agent. because a particular heterologous protein, you can express a small portion of it. You have understood that is the important protein-protein interaction motif which is required for stabilizing your regulatory protein. Then it will be much easier for harvesting this protein from the expression system. So this is a very important science which has developed with the recombinant DNA technology. So, and then another important thing is that sometimes, cytoplasmic expression is very easy. Then you don't need, any particular signals, or you will get your protein in high concentrations. But sometimes what happens is, as I told you, there may be inclusion bodies. And then there will be, there will be no disulfide bonds made in the bacterial cells. And the protein not so stable. But then sometimes what people choose is, instead of expressing in the cytoplasmic portion of the bacteria where maybe it's making the inclusion bodies, they try to modify the gene sequence in such a way that it, as soon as it is expressed, it goes into the plasm. And then they isolate the protein from the periplasma. But then it has its own advantages and disadvantages. The most important disadvantage is that the protein concentration which you will have will be very low because of the inefficient transport into the periplasm. And then advantages are many for the protein which is giving problem when expressed in the cytosol of the bacteria. By putting it into the periplasm, will improve folding, so no inclusion body formation, and disulfide bridge formation may be enhanced by the presence of DSBA and DSBB proteins which are present in the periplasm. And then there are fewer proteins, and possible leakage to growth media may facilitate purification. So, and there is less protein degradation. But these choices are when you're facing issue with the expression of the protein. But most proteins, they get expressed properly in the cytosol. Only very few of them will give this problem of inclusion bodies. Means that they won't be soluble when purified. And these problems we have already discussed. But then the glycosylation and post-translational modification like phosphorylation and amidation won't happen in bacteria. But solutions are that you proper suitable E. coli strain, and then sometimes what happens is that now with the modern technology, there is self-free systems for treating your. People are making biochemical milieus. You express a protein in bacteria and put it in a milieu where it will change it, cell-free glycosylation, phosphorylation, amidation, So various things are being studied, and so that after easy expression of protein in bacteria, one can change it in the in a biochemical environment. So I have already explained that how protease deficient BL21 strain one can use, and then thioreductase or glutathione reductase double mutants may give disulfide bridges for all. So like that, what people have many options now. So this is just a summary which says that the characteristics of suitable induction sensitive promoters, which is the most important thing for expression of protein in any system, is that it should be high, tight regulation, no leaky expression, simple and cost-effective induction. And for basic research, IPTG, tetracycline, or chain temperature is used in bacteria for induction of expression. In mammalians, I told you that it is constitutive, you don't have to put any induction. For industrial protein of therapeutics, they can use thermal, chemical, or nutritional differences for the expression of protein, so that one does not. So this is just another detailed thing for which we have already seen in another, but the important point here is that when you have this protein expression in bacterial cells, there is automatic in vivo cleavage. So when you get the extract of the bacteria for isolation of the protein, you will get your protein in the isolated intact. This is intact fusion protein. So it is automatically cleaved from the other proteins. If there are anything, it is a independent protein which is expressed. It's not that it is in a vector, so you'll have to cleave it or anything like that. You don't have to do anything. The protein is automatically just there. But you can cleave the if you have it as a fusion protein, then if you have any particular strategy to cleave it, like proteolytic cleaver or any other method of cleavage, then you can have the cleaved protein, and you just have to isolate it, and that's it. And these are the various chaperones which so that is what I said. It is not just the proteolysis that you need the co-expression of molecular chaperones, but it is also that if you just express the protein, it aggregates, and that is also kind of inclusion bodies, that the protein gets into a very odd folding process, and so it's not really dissolvable in normal functional fluids which we want to use for studying or any other purpose. So like this, you can have various different kind of, GrowEL, GrowES, or many other modifications you can do to get this. So this is just a summary of the cloning platforms. Now in the multiple cloning site, we have restriction enzymes, which are also called restriction endonucleases. And then another enzyme which we use is ligase. This is a general cloning, kind of components. So by making your gene ready, you can buy PCR-based methods, or there are other site-specific recombination methods, or there are Golden Gate and other part molecular construction methods. So there are many advanced ways of doing it, but this is just a basic slide where you see that DNA ligase can, when you are cloning and you have a vector and you have your gene of sequence. So ligase is very simple ligation. So these are the key enzymes used in this method. So if you see various different restriction endonucleases, so if you see when you are cloning your gene, you can choose any of these sites. So you will have to put the sequence ahead and back of your gene. So when you cut this with this enzyme and put the gene, and then it will be cloned in the CoR1 site or BAMH1 site, or whichever you have chosen. So the same enzyme you will use to cut it off when you want it cloned in, say, you choose to clone it in some other vector or in another setup. So it is very easy. These are the engineering cuts and pastes where you just use this endonuclease, simply put your isolated plasmid with this enzyme, and it will release the gene. Then you have various kind of selectable, bacterial selectable markers, genes, and these are the antibiotic resistant genes. So these are various different things which are used for bacteria because there it is required, and these are the antibiotics for this. So when you put an antibiotic, this will be very important. This gene will get activated when cloning is done. So when you put the antibiotic, this system will not be now toxic, and in the cloned one it will not be. And then so people use ampicillin, tetracycline, or kanamycin, or streptomycin, chloramphenicol. Like this, you can use various different selection markers. So these are various different other expression vectors than the mammalian ones is the yeast, where you can use the Baker's yeast, Saccharomyces cerevisiae or Pichia pistores. And then you have another system of insect cells where you can go very big. In both the cases, you can have very big fermenters and large scale industrial expression of proteins is done in this. So in case of this Pichia, there may be some problems and some advantages. So one has to see suits your protein. For some protein, production level is low and they get hyperglycosylated. So more than 100 mannose residue in N-glycosylation is taken as hyperglycosylation. So in this case, they seems to be hyperglycosylated, and that's why if you express, say, FSH or LH gonadotropins, you will see that their activity will change. Hyperglycosylated H will give a different response than the normally glycosylated, and low glycosylated will give a different. So functional hormones like gonadotropins and others you need, or receptors maybe sometimes, so you need to have this, but if you after it is not giving the proper response, it means that there is a problem. So sometimes secretion is not good, protein stacks in the cells, and as services this large amount of ethanol which is toxic to cells and affects level of production. Now this is about as services. Now if we see for Pichia pistores, highly efficient promoter is there, and then tightly regulated alcohol oxidase. So then this does not produce any ethanol. Very high level density is there, cell density and secretion is very efficient. So Pichia is preferred more than the S cerevisiae for, in fact, I think one of the Bangalore groups, they to express hormones in Pichia pistores, and they had certain successful attempts also. So in so Pichia yeast, which is single celled, and they behave like bacteria, but then they have, as you know, they have more advantages as having no alcohol and having all the post-translational modifications which are possible in this. Because of short of time, I'm not explaining more on this, but because there are others which are need to explain. So now here there are these are called retroviral vectors. These are for the mammalian expressions and things. Now these are RNA viruses. So now these are very important for doing human and animal research. And my field of research is that. So in this, any foreign or mutated host gene can be introduced, and now this gets easily when you want to make stable transfection of gene in human cell lines, or so then here it easily, when you use retroviral vectors, means you are using retroviral components for cloning, they easily get integrated into the host chromosome. So that is why these are good for the studies to do in human. When they make retroviral vaccines, I don't know, those proteins will go and then they will get inside our genome and what. So then this is the cosmic vector which I showed you earlier, and this takes 45 kb, very high. If you want to study a large portion of the DNA, then you use these bigger clones. And so then there is the one which is the bacteriophage lambda. Now this bacteriophage itself says that these are the viruses of bacterial host, and these are also kind of structures which are typically like a phage, and head, tail and tail fibers. So these lambda viral genomes, they can have very efficient cloning of genes, and these are 48.5 kb linear DNA with 12 base single-stranded DNA, sticky ends, both ends, and these ends are complementary in sequence and can hybridize to each other. So it is called the cohesive ends. So, and then these are also very easy for the infection. So bacteria can easily be be infected, and then this DNA circularizes at the cause site, and lambda begins its life cycle in E. coli host. So these are also want to use, and sometimes the next step which I am coming to is the expression libraries. So now these kind of vectors sometimes are used for building expression libraries when you want to do the work relating to the expression, discovering the new proteins and expressions. Then sometimes these are these can hold a longer DNA sequence. But the most important system which is used for discovery as well as for the expression of cells is the baculovirus system. People want to do screening of, they make synthetic peptides, and now they want to screen whether it is monoclonal antibody kind of nanobodies or peptides of other types. What people do is that they clone into baculoviruses, and now these baculoviruses can easily be, they call it biopenning. Now these are why they are more popular when you want to do discovery of new binding partners or new therapeutic, is because these can be, very easily, handled, and infections are very fast and they bud off, and then they, kind of, they self-replicate to the extent that you get very good yield, and you can do the, transformations of the cells, and so the host cell, insect cells, can be from the Spodoptera frugiperda or Bombyx mori, and this vector is originated from the Autographa californica multiple nuclear polyhedrosis virus. So now this is basically a virus vector, but it is having a promoter, which is the polyhydrin gene promoter, which is a viral promoter. So which can, easily can be expressed in the insect. The baculoviruses are viruses of the insect cells. So they already have the cassette for the expression of these proteins through these vectors very easily in the insects. And one can, so there are several popular expression libraries made in baculoviruses, and even for the nanobodies or for monoclonal antibodies or for so many other therapeutic discovery stage work, people have made several baculovirus libraries, expression libraries, where you just make a large scale screening of these, and these screenings can be done in 96 cell plates, or it can be done on the, for the bacterial expression libraries, you normally do it on the plates, and you soak the Whatman sheet, circular Whatman sheets are there of the, those plates where we grow the bacteria, 10 centimeter plates. So you just have those circular nitrocellulose membrane, you just soak them in IPTG, put it on that plate where bacterial colonies are. So in bacteria, it is done like that. And what will happen is by morning, each colony will express protein, which will go and stick to the intracellular membrane as a small dot. It comes beautifully. I am not able to show you I did it in Hyderabad only in CCMB. So it was very, very interesting to see that how immaculate these procedures are, and so neat and beautiful dots you will get, the Western blotting on your nitrogen for finding the specific protein expressed by which colony. So similarly here you can do it in 96 well plate format, and none of these procedures are complicated or anything. You need simple culture conditions, and they just sound very sophisticated. But if you do it in the laboratory, you don't need too many sophisticated. So come back to bacteriophage again. All this is very familiar during the school books, and bacteriophage are preferred means for expression because of the lytic cycle of bacteriophages. So for bacteria, one can use these, and for insect, one can use the baculovirus. And so these are two popular systems. Okay, so this one is the one example I just put it for isolation of these proteins. So now these, you see, this is his tag, and this is your protein. So it can easily be affinity rift on the agarose gel with the particular, or if it would have been GST, you will have glutathione bound agarose, and this glutathione will bound GST, and then when you want to elute it, you will just elute the GST solution. So very simple procedure. And so if we see the overall what we discussed today, and try to understand what is going on. So most protein therapeutics currently on the market are recombinant, and hundreds of them are in clinical trials for therapy of cancer, immune disorders, infections, and other diseases. Now, these new engineered proteins, they include bispecific monoclonal antibodies or multi-specific fusion proteins, monoclonal antibodies conjugated with small drugs specifically for cancer and certain other immune disorders, and then proteins with optimized pharmacokinetics are currently under development. So this is the most important science of personalized also or pharmacogenomics, where if a particular person has a particular defect, he can go and if get a particular kind of therapeutic developed for particular disease. If so, in last several decades, there are no conceptually new methodological developments comparable to the genetic engineering. That is the most amazing thing which I feel. And this has been most contributing technology for developing the therapeutic proteins or peptides or antibodies or whatever. It leads to help the genetically affected individuals because in those cases, no, although gene therapy is again coming into picture, but then we know that that is still a little far off. With certain technology of CRISPR-Cas and other things where we can go slowly into the genome, and there, if that will be possible, then maybe it will be important for the genetically affected individuals. So briefly, in a few, I can just tell you the kind of technology which I am using for my research, which I did not include here today because it will be important to discuss all basic concepts, other things, is that when we express the hormone receptor, FSH receptor, LH receptor, insulin receptor in HEX cells, they just automatically express. And then you can check that how the expression of these receptors can affect the function of the other receptor. And that is how we found that FSH receptor and LH receptor, they dimerize. This heterodimerization of FSH receptor and LH receptor that leads to the inactivity of FSH receptor. And this is something which may be happening in case of PCOS patients where LH receptor is overexpressed. So at this moment, I'm working on this effect of higher expression of LH receptor on the functions of FSH receptor and insulin receptor. So this is very interesting that just the expression, higher expression of LH receptor can cause so many problems. In the LH receptor and or FSH receptor, thereby it is affecting the metabolic responses of FSH receptor as well as insulin receptor. So what we need is in today is a therapeutic for neutralizing the higher LH receptor. So where I am at this moment is in France. We have a collaboration with a group where they have a collaboration with the companies interested in nanobodies. So now here the group has found 2 or 3 nanobodies which bind to the receptor and LH receptor. And I am interested in that how these nanobodies can be used for patients.
I'm sorry for the interruption.
I finished. Yes. I finished. This is the last
Okay,
sentence.
Okay, go ahead, ma'am. Okay, ma'am. Thank you.
Already at the last sentence. So that is what we feel that proteins can help genetically affected patients. And I think more research in India should focus on that. I don't find too many groups working in these areas. And that is and once again, I thank you all for inviting me for this talk. I'm very happy to take any questions if there are.
Yes, the students want to interact with you, ma'am.
Yes, sure.
Yeah, new recombinant plasmid. Can we get pattern for that, ma'am?
Be louder.
The voice is not clear. If someone can speak.
If we construct a new recombinant plasmid, can we get pattern for that, ma'am?
Yes.
Thank you.
Oh, definitely. That's what the companies are earning so much. Yes, definitely. You can make a company.
Any other questions?
Any other question?
Thank you, ma'am. Next, I'd like to call A. Hemashri Bhagwan of Second NLC to end session 8 with vote of hands.
Good evening, everyone. I welcome you to vote of thanks on behalf of each and everyone present here. To all our chief guests, Professor Rita Singh, thank you for spending your valuable time with us and making this workshop more meaningful by sharing your knowledge of the topic. Recombinant technology is therapeutic products. Even though we have rough idea about the recombinant technology, we have learned more info about it, especially about the major problem of eukaryotic gene activity and the type of expression system like insect cell, bacterial virus, and the advantage and disadvantage because according to the host cell we choose, and the bacterial host BL21T3, which can grow in research. Today we had opportunity to hear your thoughts, and I'm sure this will surely going to enlighten our path in the future. Thank you.
Thank you very much, Professor Muralidhar and everybody.
Participants are requested to maintain silence, please.
Kanji Shri Krishna College of Arts and Science representative.
Please don't make noise.
Now we'll begin the valedictory function. Before beginning the function, I'd like to request S. Tanupriya of 3rd PSC to kindly begin the function with a prayer song.
प्रभो गणपते परिपूर्ण வாழ்வருள்வாயே प्रभो गणपते 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 आदिमूल गणनाद गजानन अर्पुत तवल स्वरूपा देव देव जय विजय विनायक हिमय परशिव दीपा प्रभो गणपति परिपूर्ण வாழ்வருள்வா प्रभो गणपति 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 சாந்து வணங்கி துதி பாடி ஆடி உந்தன் சன்னதி சரணடைந்தோமே சாந்த சித்த சௌபாக்கியங்கள் யாவையும் தந்தருள் சர்க்குரு நீயே பிரபோ கணபதி கணபதி கணபதி
I request everyone to please settle down. Thank you, Ms. Tanupriya. Next, I'd like to call upon our Honorable Principal Sir, Dr. K.R. Venkateshan Sir, to give his welcome address.
Most respected and esteemed Professor
Are we audible, sir? You're connected, sir. You're connected, sir.
Yes, sir. Good evening. Good evening. Good evening. Good evening.
Okay, sir. The function has started, sir.
most esteemed and honorable and distinguished professors and academicians on the dais, most respected Dr. Elumalai, Registrar of the University of Madras, who has joined us online, faculty members from various colleges and students from various colleges and universities, dear faculty members and my dear students, I am very happy and I feel privileged to propose the welcome address today for the valedictory function for the workshop come lecture on principles of biotechnology, sponsored by Indian Academies of Sciences, and I hope you would have had wonderful deliberations and discussions with various eminent scientists and personalities and academicians from all over the country in the august presence of the great scientist and motivator and source of support and guidance for the young scholars and scientists, Dr. Muralidharan. He has been கற்றோருக்கு சென்ற இடமெல்லாம் சிறப்பு. This is the axiom, true to the axiom. He was interacting with almost all the faculty members and the students, and even today he spent more than 2 hours in our laboratory discussing with the students, and he has taken great efforts in bringing scientists and academicians all over the country, including reputed vice chancellors. Thank you, sir, for all your efforts for bringing eminent scientists and personalities and scholars throughout the country for this wonderful workshop. And it is the saying by Francis Bacon, reading maketh a full man, conversation a ready man, and writing a complete man. So reading maketh a full man, conversation a ready man, and writing a full man. And true to the axiom, sir has been encouraging in publication. He has been talking about higher education, not only talking. He wants to bring the principles of higher education to the rural area students like us, like you, the college like this. And he has been talking almost all the subjects. And now, even now, he was talking about the challenges faced by the philosophical implications, philosophical problems of the scientists. In such a manner, he has been observing not only science and technology, but also the problems of the people, the problems of the society, even the mural painters, problems of the mural painters he was talking about yesterday. So the predicament of teachers, the predicament of higher education, the predicament of professors, the predicament of students, and the national education policy, how it is important for all of us, how it can be implemented. All these things, at this age, he has not yet retired. There is no retirement for scholars and scientists like Dr. Muralidharan. So such a great personality. So Selvathud Selvam, Sevichelvam, Achelvam, Selvathul Ellam Thalai, Tiruvallur says. So you have heard about all science and technology and the implementation of all these things through the experts, scholars like Dr. Muralidharan, Dr. Bhuvankrishnan, and Dr. Munuswamy. And also you are going to hear from the registrar of our Madras University, Dr. Elumalai, who has joined online. So such wonderful personalities we are having. I welcome wholeheartedly Dr. Muralidharan for this valedictory function. I also once again request him to bestow all his support and guidance for the growth of our biotechnology, not only biotechnology, life sciences department, the research and publications. Once again, I welcome you, sir, for this wonderful valedictory function. Arivinal ஆகுவது உண்டோ பிரிதின் நோய் தன்னோய் போல் போற்றாக்கடை, says again Tiruvallur like this. Dr. Munuswamy, Emeritus Professor of University of Madras, he has also been a source of support for all our teachers and faculty members, particularly the Life Sciences Department. I once again welcome you, sir, for this valedictory function. And I thank also for sharing all your valuable ideas to the students. One more Tirukural which suits Dr. Bhuvankrishnan, as far as I am concerned, is வெள்ளத்தனையது மலர் நீட்டம் மாந்தர்தம் உள்ளத்தனையது உயர்வு. True to this Tirukkural, Dr. Krishnan, retired Director of CLRI and Head and Professor of Biotechnology of CLRI and our visiting professor. As I told you yesterday morning, this seminar cum workshop is possible only because of his efforts and guidance. And he has been encouraging all the faculty members and the young students of PG for publication. He is not only motivating towards publication but also emphasizing the cultural importance which the education can give, education for personality development, education for development of our cultural values. In all these aspects, Dr. Krishnan is very much interested in, and I welcome you, Professor Krishnan, for this valedictory function, and I also thank for being always with us, particularly in the Department of Biotechnology, as your stepson, Dr. B. Ramesh, also is with you. இதனால் இவன் முடிக்கும் என்றாய்ந்து அதனை அவன் கண் விடல். As this Tirukkural says, Dr. B. Ramesh has wonderfully organized this conference with all the support of the faculty members. He knows what work to be given to the person, that he identifies the correct person for correct job, and he has done this conference and this workshop in a wonderful manner. I also welcome you, Dr. Ramesh, and I congratulate you for the success of this program. And I also welcome the registrar of our glorious Madras University. He is the professor and head of the Department of Biotechnology of the University of Madras, and he has done research in turmeric, and his publications are in turmeric, and he has also done a lot of research in developing biodiesel, and he has also been visiting our biotechnology department quite often. A lot of times he has, number of times he has visited, and we had shared a lot of things with him, and he was instrumental in bringing the biotechnology department of our college as a research department offering PhD course. We are very much thankful to you, sir, Dr. Elumalai, the registrar of the University of Madras, for all your continuous support, and I welcome you for this glorious occasion. I also welcome the faculty members from various institutions, students from various colleges, and our students of biotechnology and microbiology, biochemistry departments. And I hope you would have had wonderful discussions and deliberations with eminent personalities and stalwarts. I hope you would have been motivated and ignited by their talks and interaction in the presence of these three great stalwarts. Once again, I welcome all of you, and I wish that you will reach your home with fond memories of staying in our college, in the department with all these scientists, and you can just start your experiments with the science and also with life, with the experience which you have had in this college. Thank you very much.
Thank you very much, sir. Next, I'd like to honor the guests, and first we like to call upon our principal, sir. Professor K. Muralidhar, sir, please come up on the stage. He will be honored by our principal, sir. Next, R. Purnakrishnan sir will be honored by Principal sir. Next, Professor N. Munuswamy sir will be honored by Principal sir. Now, this is the time for certificate distribution. First, I'd like to call upon Dr. V. Sugandhi ma'am, who is the head of the department of Microbiology department. She is also the assistant professor of Global College of Arts and Science, Vellore. Next, I'd like to call upon Kokila R. ma'am, the assistant professor of Department of Biotechnology, MMES, Women's Arts and Science College. Next, I'd like to call upon Professor H. Lillian Mayer, the Assistant Professor of Department of Microbiology, Shri Akhilandeshwari Women's College. Next, I'd like to call upon the student representatives of each college. First, I'd like to call upon the student representative of Arkat Shri Mahalakshmi Women's College. Next, I'd like to call upon the student representative of Global College of Arts and Science. Next, I like to call upon the student representative of Kanchi Sri Krishna College of Arts and Science. Next, I like to call upon the student representative of Karpaga Vinayaka College of Engineering and Technology. Next, I like to call upon the student representative of Kumararani Meena Muttaya College of Arts and Science. Next, I like to call upon the student representative of MMES Women's Arts and Science College. Next, I like to call upon the student representative of Muhammad Satak College of Arts and Science. Next, I like to call upon the student representative of Pachepas College of Mins. Next, I like to call upon Assistant Professor H. Lilium Mayor Pushpalata Ma'am of Sri Akhilandeshwari Women's College. Next, I would like to call upon P. Sharmila of 2nd MSc for the overall internal student feedback.
A very cheerful good evening to one and all present here. Now I confidently say we gain more informative and a novelty from this workshop. From the SSASC section started with Professor Krishnamurthy Kannan sir about introduction to protein science. He make us clear understood about the biocatalyst and also organocatalyst in the field of green chemistry. Next section by Professor P.B. Seshagiri sir about the stem cell biology and technology for inspiring words about the stem cell and its types and how it will differentiate into the various types. Then he introduced about, he says about the embryonic stem cell, induced pluripotent stem cell, and also the CRISPR gene editing technology and cell therapy. It is a most informative thing. Then the third section by Professor Munuswamy sir about the cryobiology in life science. He gave a wonderful lecture about the cryopreservation and his project research about the cadaveric fish sperm motility and then the methods for storing the sperm. It was the new thing for us to learn. Then the final section of the post day was given by Professor Muralidharan sir about the embryo transfer technology in Buffalo. He shared about his post experience and about his PhD work. He discussed a lot about the FSH and LSH in buffaloes. It was very pleasant to know about his experience. Then today morning lecture was taken by Dr. R. Puvanakrishnan sir. He told about the getting and decreasing of leather and the enzyme such as the protease and lipases are used in the technique. This lecture on enzyme technology in leather tanning was very useful. Then the seventh section was lectured by Professor P.K. Thelma ma'am. She taught us about the recombinant DNA technology and the gene therapies. She told about the omics and the omim. She explained about the genetic disorders such as the chromosomal abnormality, monogenic disorders, and then complex disorders. We came to know about more genetic disorders and their therapies. Then the last section by Professor Rita Singh about the recombinant DNA technology and therapeutic proteins based on their molecular types and therapeutic vaccines. Thank you so much for all guest lecture to make a conference into a more interactive manner and sharing your information on your lectures. Now it is the time to thank our chairman, secretary and our honorable principal and convener Professor K. Muralidharan sir and Department of Biotechnology Aid and Coordinator Professor B. Ramesh sir for giving such a wonderful opportunity to gaining more knowledge from this workshop. Thank you, sir.
Next, I would like to call upon Dr. V. Sugandhi ma'am for external feedback.
Good evening to all. I am Dr. Suganti, HOD, Department of Microbiology, Global Arts and Science College. It takes me immense pleasure and happiness to deliver a workshop feedback. To be true, I came with my BSc final year students. When they came here, they have blank knowledge about research. I hope this workshop gave him a wide knowledge about research. I would thank for the opportunity, Muralidharan sir, for this opportunity. Thank you. And my students conveyed that they got research interest on microbiology or on biotechnology, and especially they listened the lectures of Muralidharan sir, Munsami sir, and Puvana Krishnan sir, and Thelma madam. They were impressed by the lectures given by the scientist, and they personally told me they thankful. Thank all excellent. And above all, I personally felt this is the impact and success of the workshop because from the rural area, students came to know about what is research. This is the impact we got today. Thank you. Thank you once. Sir. And all the lectures was deliberately gave the interest in young research minds to develop how to develop a research. Thank you, sir. And above all, I thank the department faculties and the students of Department of Biotechnology for organizing the transport and excellent food and hospitality. Thank you, students. You have cooperated well with the department. I thank Indian Academy of Science, Indian National Science Academy and the National Academy of Science India and Department of Biotechnology and beloved Principal Sir, HOD Sir and my department friends for this opportunity to stand before you as the external participant and to convey my feedback. Thank you one and all. Thank you, sir, for the opportunity.
Thank you very much, ma'am. Next, I'd like to call upon Sharmila Zahir Hussain of 3rd Year Biotechnology of Mohammad Sata College of Arts and Science.
I'm Sharmila Zahir Hussain, 3rd BSc Biotech from Mohammad Sata College of Arts and Science. Firstly, I would like to thank college management and the Department of Biotechnology's faculty members and students for their hospitality, and we felt so comfortable here. We came a long way without any professors' support. We were afraid that we were afraid that will it go well? Will we be able to learn? Will we be able to mingle in this group? But despite of my thoughts, we were well comfortable, and their hospitality made me glad. Secondly, I will tell that the lectures of various professors were very knowledgeable, and it was full of details. And then I thank everyone for this wonderful opportunity. Thank you.
Thank you very much. Next, I'd like to call upon Shifa Sharika MA of 2nd MSc Biotechnology, MMES Women's Arts and Science College.
Good evening to everyone. I like to thank Department of Biotechnology of Sri Shankara College for providing such a great opportunity to learn something new. It was really fun to learn all of the new concepts here. I mean, we had no idea about research, and we are MSc students and no idea about research. So I guess this opportunity actually helped us learn about whatever the research methodology, and we even got to know more about various new concepts. And I'd like to thank everyone for providing us such a great opportunity. The guest lecture professors for providing such a great opportunity.
Actually, all the resource persons, our professors, they were impressed the level of student interaction. So we have to thank you specially for that. The students made this opportunity useful. They interacted highly. So among them, two persons who asked most number of questions, they specifically asked me to honor them. So these two people, most of the questions were put forth by them. So we request our professors, we can take a photograph so that it's a way of honoring them. It will encourage all the students to interact more. And we cannot call all the students because all of you have asked questions. We have to acknowledge that as a token of appreciation, we are doing this.
Next, the certificates will be distributed to the student representative P. Sharmila of 2nd MSc Sri Shankara Arts and Science College.
I request two students for each class, our students, on the dais, please, to collectively receive the certificates, please.
Next, I like to call our chief guest, Professor S. Yermale sir, who will give an online valedictory address.
Sir? Sir, could you hear us, sir?
I request
Sir,
everyone
could
to
you hear
please
us, sir?
maintain silence.
Sir, we couldn't hear you, sir. Sir, we couldn't hear you, sir.
Hello.
Yes, sir, we could
Hello.
hear you, sir. Yes, sir, we could hear you, sir, now.
Okay. So very good afternoon. Very good afternoon to everyone. அனைவருக்கும் வணக்கம். First of all, I would like to apologize for my delayed appearance. Since I am waiting in one other meeting in Secretariat, in தலைமை செயலகத்தில் இருக்கிற ஒரு வேலையா வந்திருக்கேன். So that is why I am unable to participate. First of all, I would like to thank our principal, Dr. Venkatesan and Dr. Muralidharan, my dear beloved friend, Dr. Ramesh, to giving me the opportunity to interact and to deliver the valedictory address. Sankara College is one of the best colleges as far as biotechnology is concerned. They are having very good infrastructure instrument facilities. I have been many times interacted with Sankara College as an examiner, as a visitor, as a guest. So many capacity I have visited. Their hospitality is very good, and Dr. Ramesh is doing very good job, and under the leadership of Professor Venkatesan sir. Since Dr. Venkatesan sir is our honorable syndicate member, so we have many times interacting with him, and we had very good input from him for the betterment of our university. So since I am preoccupied and I am sitting in front of the Secretary Higher Education room for some official work, it is very urgent work on behalf of Vice Chancellor. So I came to know that the National Conference Come Workshop is a wonderful event going on at your esteemed institute called Sangara Arts and Science College. So I am extremely sorry not to physically present over there. So with these few words, I am waiting for the call from the secretary. So I am unable to take part fully as a participant or the dignitary. So on the whole, I congratulate the team of Dr. Venkatesan sir and Dr. Ramesh and his team to have a successful event. Thank you so much.
Thank you very much, sir, for your beautiful valedictory address. Now I'd like to call upon Professor R. Purnakrishnan sir to give the chief guest address.
Good evening to all of you. Respected Principal, Professor, respected Professor Muralidhar, convener of this conference, respected Dr. Yelumalai, Professor of Biotechnology and Registrar in charge, University of Madras, and a good friend of mine the last 3 decades. Maybe he is not there. Anyway, I'm addressing Professor Manuswamy, whom I am very happy to know after coming here. I know him previously. More about that, Professor Ramesh, who is the architect of this conference, so all the faculty of this department and my student friends. It's a real pleasure for me to be here. Of course, I come here very often, but this idea was mooted about 7 months back when I had a talk with Professor Muralidhar. He was telling that there is a program is there. And immediately I told him that you please look at Kanchi. Please look at Kanchi, the Sankara College, and then our experience should go together. And immediately he agreed. That is the result, that 7 months of, proceeding further and the applications, then sending again and then requesting. Finally, the science academies, they've agreed. There is a little difference. Any function, any college can arrange. But to get a program from the national science academies, it's a honor, the college, and for you also. That was possible through Professor Muralidhar, and then all the basic things, everything were done by Professor Ramesh and all of you. Because I was seeing the last, that so many days I've been coming here, they have a big team, all wonderful team of workers joining together and then supported by all our students. It's a real pleasure to see the development at close quarters. Most important is they could do so much for the betterment of this college with a new program, but the credit goes to the principal for his vision. The very, very important is that the head of the institution should say, go ahead, should give a green signal. Here he has done that. When I said that we have got a program, just go ahead. That's what he said. And the credit goes to you, sir. I'm not going to speak all about science, that it's a great day. So we have all come here to say goodbye to you in a sense. You came here, you had a good time, you interacted with learned professors. That's more important for us. What we need is that you should learn from the professors how they have come up in life by hard work, by dint of hard work, by understanding things in a proper way. I hope I'm right if I say that it's a real pleasure and opportunity for you to come together and then be with them and question them. The questioning is more important. I was able to see so many of you were coming and then questioning. It's a very, very great development, and this we should continue. For me also, it's a great opportunity to be here with you for 4 days. That I told the respected principal and Professor Ramesh that I'll be here for 4 days. You have to support me. And that I requested Professor Munswamy also. He said, no, I'll be with you. So that's the great thing is, yeah. So on this great occasion, I'm very happy to say that all the best to you and then to the Department of Biotechnology and to the Sankara College for hosting this important program. And I wish many more programs. We are planning or talking what program you can bring next year. More important, not that today we have done this program, this is not the end of the. So what next? You suggest what you want. You can even give in messages what actually you need from the learned professors, from the science academies. So your ideas will be definitely conveyed to the respective professors and to the institutions. Accordingly, we will do. So with these few words, it's my thank you for all the support you have given. Thanks a lot.
I request our Professor Munuswamy to felicitate the occasion with few words.
As rightly pointed out, good evening to one and all present here. Just observations I want to make 2 days. Whenever I always say the intellectuals, you would have seen Professor Murli there. I am one of the admirer of him. Whenever he says that he is going to attend this, I know this even before 7 months before that. I made a point to stay with him. Whenever I go there in Delhi, I get inspiration from him. My observation, this having come here, and we people are from rural. You see, my name is Munuswamy. I'm coming from Chengalpet. All the time I used to travel to Laila College. That time I got some good marks. But what do you call perseverance is more important. Sincerity is more important. Fundamental concept is instrument. We have the dignities here all developed because of the sincerity concept. I admire our professor Moralita. So it is very difficult to get a top man in science to bring him here. You are எப்பயுமே சென்னை பல்கலைக்கழகத்தை படிக்கிறதை கொடுத்து வச்சுருது சொல்லாங்க. இந்த மாதிரி ஸ்டால் வாட்ஸை நீங்கள் பார்க்குறத கொடுத்து வச்சுருக்க எங்கேயுமே கிடைக்காது. இட்ஸ் வெரி டிஃபிகல்ட். பிசைட்ஸ் நீங்கள் பாருங்கள், சாருடைய ஐடென்டிஃபிகேஷன் எத்தனை எத்தனை எஃப்என்ஏ முக்கால்வாசி யூனிவர்சிட்டியில் இருக்கும். இந்த எஃப்என்ஏ எஃப்ஐஎஸ்சி அதெல்லாம் தெரியாது. எங்களுக்கெல்லாம் ஒரு ஆப்பர்ச்சுனிட்டி கொடுத்து நான் டெல்லி போனதால் தான் ஃபெலோ ஆஃப் நேஷனல் அகாடமி, அலகாபாத் அகாடமி, பெங்களூர் அகாடமி, எத்தனை பேர் நம்ம தமிழ்நாட்டிலேருந்து எடுத்திருக்காங்க. A few. But you go to Delhi, says, this all we get inspiration from him. Then, regarding the observation of the 2-day seminar, today national workshop and academies, you see this professor, your principal, he has studied, he has given excellent preview of genesis of academies. Very really, sir, thoughtfulness, sir. Excellent introductions about the genesis. Very few. Then, the faculty here are all Ph.D. I always insist the people who have done excellent work in the university setup will be good masters. That's really credit goes to this college. Then observation, you have seen all the lectures are excellent lectures. They work for 3 decades or 4 decades. Getting the information from them is very, very difficult. You got it. We are thankful on behalf of you. I'm thankful to Professor Muralidharan sir for having made it. Of course, Bonagres are the coordinators. Many a times we discuss with them. In fact, Ramesh is my MSc Biotechnology student. I took biostatistics for him when I was those days, 90s, those days. Then observations. All the lectures, I've seen the students are coming forward asking questions. All pertinent question, excellent question. Even the expert, they say pertinent question. Very rare to see the things of that. Very rare to say. Hats off to you all the faculties. And I just little worried. Sir has traveled widely throughout India, Kurnool, all the places he visited, he conducted this one. I asked him, sir, how the response of the student? We go to a very rural place. Are they able to speak English? Are they communicating Hindi, something like no, Swami, they do something like that. But I have seen that communication of the student, that credit goes to almost all the colleges have come. They got excellent communication skill. Thus now science is developing it. So please understand the concept and do that. Then the team, they're working here, excellent. Because many a times I'm afraid. Many a times I observe, Sir has come, anything happen to default by electricity short, because we are not in a high profile world. I never call my high profile biotech. So something happened, what will other people think? What will reflect the college? I moved with you people as own your classmates or something like that, what you can think it up. So they made a good team and faculty. Okay. Then besides the introduction of the speaker, the introduction by the faculty is excellent, exorbitant. So really fortunate. It's not a long way to go. If you continue this kind of the programs, I requested Ramesh and other colleagues, and please go ahead with this, look into a national level program and try to educate our population, student community. You are the pillars of future world. I'm really happy to be here for 3 days and communicate. You have seen me all the time. I was sitting here and like you also learning. Learning is a process. If you are very confident and if you can achieve certain things, definitely do that. And the financial things automatically come off that. If you are good, if you are getting, when Professor Pandian said, if you get 89, 90 marks, we need not go for reservation, he will say. So we rise to the occasion, come to the success in your field of research, whatever field you take it. With this, I'm really happy and thankful to the college management, Dr. Ramesh and Professor Sir, for giving an opportunity to share my thoughts of my career biology and also associate with you all. Thank you so much. Thank you.
Every good thing must come to an end. I would like to call our coordinator, Dean, and Head of the Department of Biotechnology, Dr. B. Ramesh Sir, to give the final vote of thanks.
Good evening. want to convey one important takeaway message. The last 2 days you have spent here, you could have spent the last 2 days wherever you want. You could have went some movie, anything. But you chose to stay here and observe and try to connect. You keep this up. This will help you in your life. We need contacts. We need what is going on in our field. So for that contact, you have to go to the places and make some contacts for doing the right thing. Seemingly unconnected things are always connected. For one small example, I want to tell you. All these learned professors sitting in this dais. I am now HOD of this department of biotechnology, but I studied in University of Madras during the period, my MSc period. Professor Munuswamy taught me biostatistics. He was my teacher. And Professor Poonakrishnan guided my MSc thesis. And our principal, beloved principal, he taught me English during my BSc. So all these three gentlemen are my teachers. So Professor Poonakrishnan, Professor Munuswamy, I came to know about Professor Mulli. So this relationship is beyond 20 years, as you see. So similarly, we are ready to help you. We are ready to encourage you. This bond should continue. So you keep on attending this kind of workshops. This should be your takeaway message. You continue to do this and appear for this kind of. So with this, I wish to deliver my vote of thanks. First, I would like to thank from my core of the heart for Professor K. Murlidhar. He is the convener of this program. So he acted as the convener. He planned all the things and he invited all the lectures. Most of them unknown to us. Invited them only because of him. The IASC professor gave 2 lectures. This is very rare. He couldn't even pick our call, but he gave 2 lectures, 2 hours spent. So, sir, thank you very much for your services, sir, as a convener and as a mentor. And I seek your blessings and to continue the mentorship. Thanks, sir. Next, I would like to thank our Professor Poonakrishnan. He is the main reason, as he explained very well. It was the idea became reality in the past 7 months. So for all his effort and continued guidance, all other persons came at the evening, but he came at the morning itself. So he spent the entire day seeing all the preparations and everything. He encouraged our students. So thank you very much, sir, for your support and mentorship. I thank Professor Munuswamy. Actually, I asked him one day, but he volunteered that he will accompany with Murlidhar sir and he will be here at all 3 days and mentor us. So thank you very much, sir, for your. Actually, his daughter is there, but he came here. He is from faraway place. He should be there in the home, but he is here with us. Thank you very much, sir, for your sacrifices and everything. I would like to thank Professor Yelumne, Registrar, University of Madras, and HOD of Biotechnology, University of Madras. Even though he is not here, whenever I ask him anything, he will tell, okay, Thambi. So immediately he will accept. Likewise, he immediately accept, and even in the evening, sir, can we change this into online? Okay, Thambi, I am ready. So that kind of support he is giving. Thank you very much, sir. And I would like to thank all other speakers, Professor Krishnamurthy Kannan, Nagaland Central University, Professor P.B. Seshagiri, Indian Institute of Science, Professor B.K. Thelma, University of Delhi, Professor Rita Singh, University of Delhi. So all these members shared their knowledge with us. We sincerely thank all the members. And I thank our management, chairman, secretary, and our beloved principal, Dr. K.R. Venkatesan, who supported from the start of this event. This program is supported by Academies of Science, but even before receiving the money, he said, I will support you. The college will support you, and he stood by his word. So he provided all the facilities. Everything we use is given by our principal. Sir, my wholehearted thanks to you, sir. and we seek your continued blessings and support. And I thank all the Academies of Sciences because they are funding this program. Their purpose is to encourage students. I hope it is done here. So I thank all the Academies of Sciences and colleges, the participants, the heads and staff members from other colleges, particularly Dr. V. Suganti, HOD of Biotechnology, Global College of Arts and Science, Ms. R. Kokila, Assistant Professor, MMES Arts and Science College. You both stayed back all these days, and heads and staff of our college. And I thank Dr. G. Suresh, HOD of Microbiology, for being part of our team. Actually, even though his other department, he became part of our department, and he is being part of our organizing team. My sincere thanks to you. And this is also networking. He is my student. So this is second generation. So this is networking. And I sincerely thank the organizing committee, our staff members and volunteers. Particularly, I wish to name Dr. V. Guruchandran for publicity, overall coordination, everything. Dr. P. Saravanan, student enrollment, for all his technical support. Dr. E.G. Jagan, food committee and all other hospitality. Dr. D. Chitra, for all the proceedings, reports, invitations, everything. Dr. A. Shyamla, the entire event management. Dr. S. Malati, organizing the sponsors and the technical help. Mrs. A. Rajalakshmi, for technical assistance. And our sincere non-teaching staff members, Venkatesan, Kasi Vishwanathan, Yelumalai, and all other members of our college, particularly electricity members, they are with us 2 days. He is talking about stem cells. So I thank the electricity people, and I thanks sir. And I thank transport, cleaning crew, all the staff members of our college, and all the members of the subcommittee, all the students, because student power is the best power. They utilized you. Thank you so much. I thank all the media personnel, particularly Dinamalar. He published today itself the article. I thank all the sponsors in and around Kanchipuram. Above all, I thank all the participants and your positive feedbacks. So I thank one and all. If I left anyone, that also to be covered. So it is not intentional. I thank one and all. Thank you so much.
To conclude this 2-day national workshop, we like to end it with a national anthem. I request you all to please stand up.
भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उत्कल जलधि तरंगा तव शुभ नामे जागे तव शुभ माहे गाहे तव जय गाथा गण गण मंगलदायक जय हे भारत भाग्य विधाता जय जय हे जय हे जय 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 हे
Thank you very much for attending this workshop. Hope everyone has learned something new, and this was one of a kind experience. Thank you and have a good day.